Hello, everybody. It's really my pleasure to be here with you again for March 16th VanBug event. So to start things off, as usual, so this is going to be our schedule for today, and we're running 5 minutes late. So I'm just going to start off by introducing the speakers, our student speaker and our main speaker, Dr. Aline, and then as well, I'm going to be talking about our sponsors, which are, as usual, VanBug is sponsored by the Bioinformatics program, graduate studies program in Bioinformatics, and by Genome BC, Ecoscope program at UBC, the Canadian Bioinformatics workshop, and we are also glad to be to announce that we're sponsored by Connexia Health, and Langara College, and Stem Cell Technologies Research, the company. So one important thing is that, of course, VanBug has been affiliated with the ISCB, the International Society for Computational Biology, and we do have two sister organizations, which I invite you to check their websites. They have monthly talks, just like us, Monbug and Torbug. So with that, I'm gonna just introduce our speakers. So first, we're gonna have Angela from the Joy Lab at UBC, and Angela will be talking about the work on geographic pipeline for SARS-CoV-2 genomic surveillance. And our main speaker today is Dr. Aline, and I'm quite excited for her talk. So Dr. Aline is an assistant professor at the Department of Obstetrics and Gynecology at Faculty of Medicine at UBC. She's the director of Science and Informatics at OVCARE, which is British Columbia's ovarian cancer research program. And she is alumni of UBC, so she had her PhD in statistics from UBC in 2013, and since then she has been working on predictive models for improving patient care, especially in women's health and oncology. And her focus also includes topics such as ethics of data sharing and privacy concerns, especially in this digital age, and its implications when it comes to health and AI modeling. So I'm really glad that we're having these talks today, and I'll start off by giving it to our introductory speaker, Angela. So Angela, yeah, if you'd like to take questions throughout, if the audience have questions, please post them in the chat. Angela, feel free to take the questions at any time that you want from the audience, including if the audience would like if somebody in the audience would like to speak up, then please maybe have that at the end, so it doesn't interrupt the flow for your questions. So I guess, yeah, I'll hand it over for you, Angela, and then I'll take it back.
Okay, I'm just waiting you to end your screen share so that I can start mine. There we go.
There you go.
Great.
Okay.
Hey. Hello, everybody. My name is Angela. I'm a third-year PhD student in bioinformatics, and I work at the Jeffrey Joy Lab and the BC Center for Excellence in HIV/AIDS at St. Paul's Hospital. Today, I will be briefly discussing our phylogeographic pipeline that we've developed for SARS-CoV-2 genomic surveillance in Canada. Right, so phylogenetics enhance our ability to survey epidemics, essentially by modeling viruses' evolutionary divergence over space and time using evolutionary trees. This can reveal the underlying epidemic dynamics. So, for instance, we can estimate divergence times and the origins of different lineages. We can identify the emergence and spread of lineages or groups of mutations, as well as discern any differences they may confer in terms of virulence, transmission, or immune evasion. And this has become very popular, I would say, with COVID-19 in terms of the wide use of pangolin lineages such as B117, as well as next-strain clades and GISAID clades. And within phylogenetics, phylogeography is the study of the principles and processes governing the geographic distributions of lineages. And this type of approach, we can try to understand relative contributions of different transmission sources, as well as to evaluate the effectiveness of different non-pharmaceutical interventions on viral dispersal. So with that, our main objective was to develop and apply a phylogeographic pipeline to characterize SARS-CoV-2 importations and dispersal dynamics in Canada in order to evaluate the effectiveness of border restrictions and flight bans on COVID-19 burden. So today I'll be briefly discussing the methodology we've developed for this, and this has been pushed to a repo on GitHub entitled SARS-CoV-2 Canada 2020, based on our analysis for the 2020 epidemic. And so within this repo, each subfolder is a build, if you will, with a new data set. And within each build, there's a workflow that I will be discussing today. And this starts with data cleaning and then subsampling, inferring trees, inferring the states within that tree, and then some downstream GIS and timeline modeling. Today, I'm really just going to be walking through up until the state reconstruction step. So all of these analyses are based on the publicly available GISAID repository of SARS-CoV-2 sequences, which has become very large. Earlier in the week, it was almost 9.3 million sequences of around 29 KB each. So this is around 300 gigabytes of just the FASTA file alone. So this has necessitated doing this on an external hard drive. So the first step is to subset this on the basis of collection dates within the range of interest. Then the first cleaning step, which is to remove any sequences that are not from humans, that are epidemiologically linked, as according to previous analyses, known temporal outliers that may be due to sequencing errors, incomplete dates, with the exception of Canada, because we want to use that data, and if the sequences were on the next strain exclude list. Next, we align the data, and we do this using the viral MSA Python script that was shared by Nima Mosiri on GitHub, and this is essentially a Python wrapper of the Minimap2 alignment, and we align to the Wuhan Hu1 reference. Then another cleaning step, which is to remove sequences with more than 20% ends or ambiguous nucleotides or greater than 10% gaps. Then we mask the alignment. So this has been sort of discussed in the field pretty widely, and essentially there are a number of sites in the genome, such as the untranslated ends and a few sites known to be homoplasic that are not used for phylogenetic inference, so we mask those. Okay, now that we have a clean alignment, the next step is to subsample, and we do this for computational, easing the computational burden, as well as reducing the sampling bias towards countries that have more sequences per case than other geographies. So yeah, we've developed this step, and essentially it's subsampling Canadian and global sequences proportionally to either provinces or countries' contributions to monthly cases over the study period. So essentially the probability of selecting any given sequence from a given geography is proportional to its contribution. And we use the case counts over time from the R coronavirus package, which is useful. It pulls from the Johns Hopkins data. And for the Canadian cases, we use PIAC data. I've also developed sort of an add-on to this where you can either choose to take all of the Canadian sequence data available or subsample it to any number of percentage of what's available. And we had to do this just because Canadian labs have been sharing more data. So whereas previously we needed to keep everything, now it's actually too much for the computational burden. So we sample some percentage and then take the remainder of global sequences up to a total of 50,000, which is sort of the computational limit-ish of one of the subsequent phylogenetic inference steps. And then for each strategy, we take usually 10 bootstraps. And just to sort of justify the subsampling strategy, showing here the cases over time, as well as the sequences available and the total sequences subsampled. So on the left column, this is the global context sequences. So in panel B, you can see, for instance, this big blue smear. That's the UK, which has been the country that's shared the majority, or like been the best at sharing their sequences. So they're overrepresented, and our subsampling approach decreases their representation in the data set, as you can see here. And it just normalizes this relationship of cumulative cases to cumulative sequences subsampled. Okay, now that we have our subsampled alignments, we can build some trees. So the first step is that we infer approximate maximum likelihood trees in FastTree using a GTR model. Then we root using outgroup rooting on Wuhan Hu1, the reference sequence. Next, we use the Tempest tool to estimate a strict molecular clock of divergence over time, as well as calculate the residuals of that fit. And we use those to exclude temporal outliers. So if a sequence has absolute value of its residual further than 3 standard deviations of residual away, then we remove that sequence, as well as remove any outliers that have the pendant edge or terminal branch length greater than 12 mutations, which is more evolution in a given person than we would expect. And this just cleans up the signal in the phylogeny. Next, we pull dates. So this is a necessary step to get sort of a TSV file of all the dates corresponding to all the sequences and trees. And this, in addition to the trees, are all fed into LSD2, which is a time scaling tool within the IQ-Tree2 suite of tools. And yes, we go from substitutions per site to the units of time, as well as infer incomplete Canadian dates, which sometimes are submitted to GISAID. And we do this on our local cluster of our group. So typically I set this up across 64 threads, and it still takes around 12 to 16 hours per tree, with the added computational burden of inferring those incomplete dates. And just to show sort of what that temporal outlier cleaning looks like, on the top 2 panels, you can see the residuals and pendant edge exclusions that are made, and the pre-clean and post-clean molecular clock signal. You can see that we have an improvement in the R² value and just a general better fit. Okay, now with our time-scaled trees, we go into the actual phylogeography component, which is inferring the ancestral states across each of the internal nodes based on what the tip sampling country or province was. And we use the APE ACE tool for this. And then using this output, we identify Canadian internal nodes that are preceded by a non-Canadian internal node, and we deem these Canadian sublineages, as well as we identify the most likely parental state of each Canadian tip. So what was the most likely direct transmission source of that tip? Then with this output, we make some nice colors. We pull the inferred LSD dates for the incomplete tip dates. And then the bulk of the analysis analyze states where we identify sublineages and singletons as rolling importation rates over time by place and lineage. We develop a sublineage nomenclature system. We summarize across bootstraps, and we quantify the relative contributions of different transmission sources over time. And we do all this to evaluate the effect of different non-pharmaceutical interventions on these rates. So just to clarify this one point, so sublineages and singletons are both introductions. Sublineages are introductions that result in sampled domestic transmission, whereas a singleton is a Canadian sample tip outside of a sublineage with an international parent state. So it resulted in no further sampled transmission, although perhaps there were descendants that were not sampled. Okay, so yeah, we applied this pipeline for this analysis as available as a preprint and is in review at eLife, early and ongoing importations of SARS-CoV-2 in Canada in 2020. And I'll just show a couple of our main results here. So this is the origins and destinations of SARS-CoV-2 sublineages overall. So we can see the USA was the biggest contributor of 52% of sublineages. We estimated around 1,700 introductions overall. And 63% of our Canadian sequences descended from only 22 sublineages that had more than 100 descendants. So we see that sort of over-dispersed type of spread that has become characteristic of COVID-19. And in terms of the effect of the border closures, we found that introductions decreased in April following border closures on March 21st, but were not eliminated. So we see this large drop on panel A. You can see, for instance, when the maximum stringency came forward on March 21st, a very notable drop in the importation rate. And although that did happen, we do see this sort of trickle effect over time. And additionally, we found that sublineages introduced later on were smaller and were detected more quickly. So it was really those early sublineages which drove the COVID-19 burden in 2020. So the main challenges of this approach are the enormous size of the data set. So our sort of solutions for this are to parallelize. We have our subsampling strategies, and restricting to particular time periods does help. Also, there have been challenges just because the GISAID data has changed over time, what metadata structure has been there. Sometimes the lineages called do change with different pangolin versions, so that's been a challenge. And also now we have differences in testing capacity and reasons for testing, and all these different components which affect the underlying basis of our subsampling strategy. So we're working on improving our case imputation to account for these differences. So yeah, I'd like to thank my collaborators for this project in our lab: Vince, Rachel, Gideon, Art, Michael, and Jeff, as well as Cancogen, which was a consortium co-author on this preprint, all of the GISAID contributing labs and Canadian Public Health Laboratory Network, as well as our funding sources, the Center for Excellence in HIV/AIDS, and my thesis committee, as well as the Bioinformatics program. So thank you, VanBug.
Thank you so much, Angela. So if anybody has questions, please put them in the chat. And I guess while we get any questions, maybe I'll ask first. Do you find that you had a problem with, so you were saying that you were trying to basically get the sequences to be proportional to the number of cases that are reported in a given month from a given jurisdiction. And I can see that that's easy to do when you do have a lot of sequences, but the other way around, where if you have jurisdictions, like for example, early on, especially in February, there were a lot of cases that are from Iran, for example, but were there enough sequences sampled from that time period that are published, that are public? And what do you, how does that affect your
yeah, that's definitely an important question. So actually for Iran and Italy early on, we acknowledged that there was a lack of sequences. So any Canadian tips that had travel history available and listed either of those countries, we actually reassigned that to be the sampling location. But that's just for those two examples, and absolutely underrepresentation is not adequately accounted for in the subsampling method. I mean, it normalizes it a bit, but if there's zero, I mean, we're not going to be able to account for that in this. There are some novel methodologies developed by Philippe Lemay's group to incorporate unsampled tips in the phylogeny for this type of inference. So it's not entirely scalable to 50,000 sequences in a phylogeny, but I think that type of approach to have unsampled tips would be definitely helpful.
Mm-hmm. I guess another question is, what is the downside of basically not sampling? If you the UK has tons of data, but then if you're building the phylogeny, I imagine wouldn't a lot of these would be just concentrated together in the same clades or subtrees. Would that negatively affect your analysis, besides it's easier to deal with less data in terms of computation?
Mm-hmm. So you're asking whether there's sort of a saturation point in terms of the informativeness of sequencing?
I guess, yeah, does it negatively, does it bias your conclusions? So is it useful just to avoid, yeah, sorry. Yeah.
So that's sort of one of the justifications for the subsampling approach. So we downsample overrepresented geographies, but there may still be some lingering overrepresentation where, yeah, for instance, you have like a bunch of identical sequences from a big outbreak that was sampled pretty deeply, and that it could bias the inference in terms of like inferring the internal node state, because if you have a lot of nodes from the UK, you're more likely to have that perpetuate deeper into the phylogeny. So I would just say that subsampling strategies are at this point the best way to deal with that.
Cool. Okay. So yeah, if anybody has any questions, I guess you can take it offline later. And thank you so much, Angela, for your talk. It was really helpful and pleasant to attend. I guess, Dr. Aline, please take it over from here. And you've mentioned that you prefer questions to be concentrated towards the end. So yeah, if anybody has a question, make sure to remember it for the end, and then you will get a chance to like speak up and ask your questions.
I see my
Yes, I can see the title slide. Yes.
Okay, perfect.
Awesome.
All right. Good afternoon, everyone. Thank you so much for having me. I'm really excited to speak this afternoon. As Bara introduced me earlier, I am a statistician by training, and I'm working right now in the Department of Obstetrics and Gynecology, which means that a lot of my research is really taking modeling tools and applying them in practice to impact women's health, and that's what I love to do. So today I'm going to be talking to you about something I'm super excited about, and it's the possibility of doing risk evaluations and screening for the prevention of endometrial cancer. So before I start my talk, I kind of want to see how much does everyone know about endometrial cancer? So endometrial cancer is cancer of the uterus. So I'd like you to type in the chat if you can, what you think some of the risk factors or the symptoms of endometrial cancer are. And so I'll just kind of pause for a minute and let you do that. So anybody who, just type whatever, I'll explain, and maybe we could have another chat, a quiz at the end to see whether or not my slides were informative. And while you're doing that, maybe I'll take a moment to acknowledge that I'm talking to you this morning from Vancouver General Hospital, where my office is. This is the beautiful view from here if you go up to the roof. This is on the unceded territories of the Coast Salish people. All right. Okay, so briefly speaking, the outline of my talk is going to be, first I'll give a little bit of background about what is endometrial cancer, who it impacts, and why we should care. Then I'll talk a little bit about how endometrial cancer arises and how it evolves. And then I'll address how it's diagnosed, and I'll make the case for why there are opportunities for prevention and screening, and they're important and we should take them. And then finally, I'll talk about RESToRE, which is our feasibility study to detect and prevent endometrial cancer. All right, so first things first, what is endometrial cancer and why we should care? So endometrial cancer is cancer that starts in the lining of the uterus. It is traditionally a menopausal disease, so that happens more later in life. So 85% of women diagnosed with endometrial cancer are over the age of 50, fewer between 40 and 50, and less than 40 is only about 5%. It's the most common gynecological disease of the developed world. It has the best survival outcomes of all the gynae cancers, but it also has the highest number of deaths, and it's mostly due to the high prevalence of this disease. So if we look at the world map, The dark blue here is the darker the blue, the higher the incidence. So you could see that endometrial cancer is generally known as the disease of the developed world. As of 2018, it is estimated that there are about 385,000 new endometrial cancer per year. And of those, 65,000 are in North America. So this graph shows the age-adjusted incidence rate worldwide, and we can see North America, Northern Europe, and Russia have the highest incidence rate, followed by other countries such as Australia, Saudi Arabia, China, Brazil, and so on. If we look at the incidence of endometrial cancer based on published cancer statistics compared to other cancers that impact women, we notice that it's the 5th most common overall in North America and the most common in gynecological cancer in the world, 2nd most common in the world, sorry. Now, the only issue here is with these cancer statistics is generally the way they define incidence is generally generally speaking, incidence is defined as the number with EC over a number at risk. The way they're computed in these cancer statistics is new EC diagnosis versus all females. And that's a problem because not all females are at risk for endometrial cancer. You're only at risk if you still have your uterus. So it does not account for the rates of hysterectomy. And so once you've adjusted for the rates of hysterectomy, we noticed that the numbers are really different. And there was a really nice paper in the Journal of Clinical Oncology that shows that it's actually the 2nd most common diagnosed cancer in women after we adjust for the rates of hysterectomy, so for the population at risk. So the world cancer statistics are a little bit misleading in that sense. There was a recent study in the American Journal of Obstetrics and Gynecology that looked at incidence rate over time and by age group, and they compared it to the prevalence of obesity, and you could see that, you know, for the incidence rate is rising over time for endometrial cancer, particularly in the 45 to 54 or the 55 plus age group. But also we're seeing the rates, it's kind of mirrored by the trend in the increase in obesity. And we're seeing in the younger than 45, a really large trend in obesity, which is kind of an indicator of what's to come in the future for this cancer. So we can really expect this to be rising. And the prediction rate is that it's gonna, the rates are gonna double over time in the very near future. And so this is a really big problem. Not to mention that it has the highest number of deaths, again, as I said earlier, because if we look at the rate of death, it's not very high because it's the most common malignancy, but at the same time, if we look at the absolute number of deaths, it is the highest in all gynecological cancers. So physical inactivity and BMI are accounting for 40% of all the attributable risk of endometrial cancer. So that's the biggest, one of the biggest drivers here. And there are different types of endometrial cancer. It's a very heterogeneous disease that differs in origin, etiology, and outcome. So for the longest time, we've thought of endometrial cancer as 2 types. So there was, this is a paper from 1983 by Bachman that sort of classified it as 2 types. So the type 1 is histotype endometrioid, and it's usually low grade, very indolent, associated with obesity, proliferative endometrium, and better survival rate. And type 2 has been thought to be the less common non-endometrioid type, very aggressive, associated with old age, present in atrophic endometrium, and has a worse prognosis. But, you know, there has been lots of studies that have sort of shown that, you know, you could actually find both in the endometrioid, you could find atrophic endometrium. So you could find patients in both, and they're not really very good discernation between type 1 and type 2. And new molecular studies, including work that's done in our own lab, has demonstrated that actually our modern understanding of the molecular classification dictates our modern understanding, and it has been further defined, but this has not really been studied yet epidemiologically. But if we kind of stick to endometrioid and non-endometrioid types, which are found in cancer registries, the histologies that are most common found in cancer registries, we could see that once we start comparing the incidence between hysterectomy correction has an important impact on trend, and we can see the trend attenuated after correcting for hysterectomy rates overall. And when we start looking at the types of endometrial cancer, the trend within the endometrioid type is pretty stable over time, but the non-endometrioid type, in contrast, has been rising significantly even after we correct for hysterectomy rates. And then if we further take this non-endometrioid type 2 group and we look at it by ethnicity, we see a huge racial disparity. So here, again, this is the study by Clark et al. They looked at the various increase in incidence rate by ethnic type, and then you could see the non-Hispanic Black has the highest rate of non-endometrioid endometrial cancer, which is, if you recall, what we call the more aggressive type. So this is a big problem. Now, to really understand this, we need to look at how endometrial cancer can arise and evolve. So here's the menstrual cycle. So in the follicular phase, there's a surge of FSH, which induces the development of a follicle. So this is a female menstrual cycle. Then estrogen produced in the developing follicle will result in proliferation and the growth of the endometrium in preparation for pregnancy. And then during the luteal phase, during the luteal phase, estrogen continues to be released by the corpus luteum, but there is a surge in progesterone, which happen in the which isn't sort of there in the follicular phase, but is there in the luteal phase. That sort of kind of prepares the endometrium for, implantation of the baby, and so that they could this balance that's produced by the progesterone counteracts the effect of estrogen, and then if there is no implantation, this results in menses. So cancer arises when there is imbalance. If there is excess estrogen, that's not opposed by progesterone. But now you think, okay, so this is in menstruating women, but I told you earlier that most of the women that get endometrial cancer are not in their postmenopausal often, and they're not menstruating anymore. So how do they get this excess estrogen? Well, in menopause, all of this the production of hormone ceases, and so progesterone is not there to counter estrogen, but estrogen should technically not be there either. But what happens is, if let's say you have exogenous estrogen or endogenous estrogen from other sources in the body that can be produced, then you will, and without the presence of this progesterone to counteract its effect, then you have this estrogen dominance or this imbalance. So estrogen is known to be produced by adipose tissue, for example, which provides growth signal to the endometrium. And especially in menopause, it's unopposed by progesterone, and that causes the problem. And if we look at how the cells look, so the majority of endometrial cancers start with a proliferation of the endometrium the proliferation doesn't stop as discussed earlier because it doesn't have this, it has this imbalance. And then from proliferation, we could see crowded endometrial glands that are endometrial hyperplasia, which then become more and more crowded and develop into atypical cells or endometrial intraepithelial neoplasia, which is a precursor to endometrial cancer and is found 50% of the time concurrent with cancer. And so that's how it evolves. Now, if we look at risk factors, most of the risk factors are associated with obesity, diabetes, also with metabolic disease, metabolic syndrome. There are some reproductive factors, nulliparity, infertility, age at menarche, and a late age at menopause, breastfeeding, combination oral contraceptives. So here the green are protective and the red are negative effect on endometrial cancer risk. Physical activity improves risk. Obviously, lack of physical activity would be a risk factor. Cigarette smoking, again, it's shown to sort of reduce the risk. However, I'd suspect that this is kind of a compounder. That's basically cigarette smokers tend to be thinner, and so that's kind of confounded with obesity, I would imagine. And so these are the various risk factors that have been studied in association. Now, if we look at how on the cellular level, how the cancer arises, we see that we start by, in a normal histology, once we start seeing unopposed estrogen exposure, so that's again estrogen that's not counteracted by progesterone, this microenvironment, along with an initiating event like a genetic mutation in PTEN, KRAS, beta-catenin, etc., could sort of, and so because this proliferative environment and this initiating mutation, this sort of could lead to evolution of the mutation that can lead to acquiring additional mutations, which then becomes cancer eventually. So how it's diagnosed? In 90% of the cases, endometrial cancer patients will present with abnormal bleeding. So abnormal bleeding is defined as any menopausal bleeding or any premenopausal irregular bleeding that's kind of irregular in its frequency or its intensity. The way that it's once a patient presents with these symptoms, they are subjected to something called endometrial biopsy, so where they collect tissue from the uterus and evaluate it morphologically. This is done usually in the office by a family physician or a gynecologist. It's often painful, but it's the gold standard for detecting cancer, and that's what's currently done all of the time. Now, if we think of the progression to cancer, from normal to cancer, we see that once we have the biological onset of disease, then we start getting these mutations, and these mutations are really detectable actually before even the symptoms arise. One thing I didn't say about symptoms here is that the symptoms of abnormal bleeding in premenopause are very, very common. So 1 in 3 women have abnormal bleeding. And so these symptoms are not specific. In fact, if you look at everyone that has done an endometrial biopsy, there's less than 5% of those endometrial biopsies result in a cancer diagnosis. So you're biopsying a lot of women who may not necessarily have cancer. And so, the idea is there something, if we are able to detect the mutations that are associated with cancer, maybe that gives us opportunities to target minimally invasive detection. So if there's a way to do this without doing this biopsy, and then it's an opportunity to prevent if you're still asymptomatic. And not only that, you also have the ability to sort of serially monitor if the method is non-invasive. So you may want to test and test again every few years, and sort of it's a screening approach for screening, really. And so there has been some screening approaches that have been proposed, for example, with the Tailor brush or the PAP brush, or cervico-vaginal self-sampling, or the tampon. And again, just like with any testing procedure, what you want is you want a good sensitivity, which means that the test can identify cancer when the test is present. And you also want good specificity, meaning that the test can truly identify the people without cancer, so that you don't want to necessarily call everyone, you want to make sure that you're not over scaring people, particularly if they're asymptomatic. So if we look at the test characteristics of these various tools, so the pap smear has good characteristics that have been shown, but actually in reality, when you look at their accuracy, when other people have repeated these and found that the accuracy is only 50%, which wasn't great. The Tailor brush is really good as well. The cervical scraping was good, a little bit lower on the specificity side. But what's really interesting here, it's this last row that looks at tampon tests and vaginal swabs. And this was done by our own team here in Vancouver, who looked at mutation in a panel of 11 commonly mutated genes in endometrial cancer. And they were able to detect it. So they looked in a group of women with endometrial cancer compared to women who are healthy but of the same age. And they found that the sensitivity was 94% and the specificity was 97%. And this was very encouraging. What's most encouraging too is that these tampon tests and the vaginal swabs are, someone's able to self-collect them at home. And so they do not tax the healthcare system, and they could be kind of used to self-monitor over time as well, if symptoms persist and that sort of thing. There has also been work that's been done on the microbiome, where they tried to look at bacterial growth in the uterine tract, and see how that correlates with incidence of disease. And basically in a recent study by Walsh et al., they found that actually alpha diversity was not that different between normals and endometrial cancer. But if you look at the beta diversity, there was a significant difference, especially in the lower tract, as opposed to the uterus. So their study, they looked at microbiome in the lower tract that could be obtained from self-sampling, say, versus microbiome of the uterus that could only be obtained during, let's say, a hysterectomy. So there was a detectable difference of the beta diversity of cancer versus benign pathologies in the lower tract. They also they profiled all of the bacterial families that could exist, and they're linked to cancer. And they found this family of bacteria to be very predictive of endometrial cancer, especially when the vaginal pH level is high. And so these are very promising signatures that could be used to identify cancer from minimally invasive tools that could be self-sampled and self-tested at home. So that's exactly what we say here. And then I think the key thing here is that these tests can be repeated, right? So that you could find an anomaly and maybe you don't have symptoms, but then a few months later could try it again and see whether or not that anomaly persists. And the idea is if you're premenopausal and you're experiencing anomalies, every time, which, and a lot of people have reported normal mutations that are found in normal tissue. What happens generally is that these mutations correct over time. So every time you shed the endometrium during your menstrual cycle, at the next cycle, it's kind of an opportunity to start fresh again. And so a lot of these things reverse and correct over time. And so being able to do this serially in the population would be very interesting. And particularly that the tools that are being used, such as tampons and feminine hygiene products or self-swabbing, are pretty acceptable and are used to screen other cancers like cervical cancer. So this kind of brings me to the next phase. So the opportunities to do screening and to do prevention in this cancer. So endometrial cancer can be reversed 80 to 100% of the time with the use of chemo prevention. So this is intrauterine device that has hormone, or for example, progesterone therapy can be used, and it's very effective 80 to 90% of the time. Also, we know that the endometrial cancer risk is very sensitive to weight loss. So a reduction of 1 kilogram a BMI reduction of 1, which is kilogram per meter square, has the potential impact of 9% reduction in cancer incidence. In addition, so if let's say lifestyle interventions are not feasible by weight loss, so physical activity and diet, bariatric surgery, so actually surgical weight loss, is also very effective at reducing endometrial cancer. And if someone is postmenopausal, they could actually just do a hysterectomy. In fact, current guidelines recommend that Lynch syndrome patients get annual biopsies until they reach menopause. And then after that, they get a hysterectomy as soon as possible. So Lynch syndrome is one, it's a familial disease, a hereditary disease. It's associated with endometrial cancer, but also with colorectal cancer. And it's the only population where screening is currently recommended, not only so that, mostly to prevent endometrial cancer, but also colorectal cancer, because that's the same, the disease is sort of associated with both cancers. And so, so this is a population that is currently getting screening. The other population that gets screening or that's investigated is the population with abnormal bleeding, as mentioned earlier. So they're typically investigated by biopsy, and if they get an EIN or cancer diagnosis, of course, the recommendation is that they get a hysterectomy or they get hormone therapy if they have hyperplasia. Now, if someone is still premenopausal, they may consider leaving their ovaries and preserve their ovarian functions. If they're still interested in having children, then they may opt for fertility-sparing options such as progesterone therapy for a period of time, and then they try to get pregnant before they go and do their hysterectomy. So those are the current recommendations. Now, why isn't prevention and screening done in a general population? So the issue here is that when we think of test characteristics, we often want to look at the predictive values of these tests, right? And they rely heavily on prevalence. So for example, if you look at 2.6% prevalence, which is the current prevalence of endometrial hyperplasia or endometrial cancer in a general population, a screening test with sensitivity of 97.6% and specificity of 90% has only a positive predictive value of 50%. This means that 50% of those with a positive test truly have the cancer. So even if we use the minimally invasive tools, which I just told you have like excellent sensitivity, we're still going to be kind of scaring half the people because they don't have the cancer, but yet we've told them that they might. So but if you look at, once you just sort of increase the prevalence of the population to a high, or increase the prevalence of a disease to 32%, you see that similar test characteristics will give you a much larger positive predictive value. And so that's the idea, right? You want to look for these populations with high prevalence. And there are three previous groups that have looked at building risk predictive models using epidemiological risk factors. And these three groups looked at very similar, they looked at similar risk factors, different subsets of them, and they've used different modeling approaches. But their idea was, and I'll try to explain this a little bit more using math, is to compute the absolute risk of developing a cancer in an age interval. So for example, the absolute risk of developing cancer between age 50 and 55. So then what you do is you look at the cancer-specific hazard rate, and you basically correct that by the competing risks. And this cancer-specific hazard rate is made up of two components. The first component is the baseline hazard. So for example, this is kind of a population-based number. So we're looking at, for example, the age-corrected mortality rate in the population times the relative risk model. And this here is a factor of covariate, you bring in covariate. So this is where the risk factors come in. So, and this could be, for example, a Cox model, which was the case in most of in the two out of the three models that were developed. But there could also be like a machine learning model, a deep learning model, right? So this is just a model that estimates relative risk as a function of covariates. So once we have this lambda 1, we could plug it in here, and here we could correct for competing causes. So the risk of dying of other reasons. So, and that's how we obtain this absolute risk. Now, then what happens is you want to plot these absolute risks for all of your population. And then you could see, for example, here in this case, you could see where everyone's risk is falling, right? And then if you look at this predictiveness curve, it's created by ordering the estimates of absolute risk from lowest to highest and plotted against the probability. So for example, at p equals 0.9 here, the risk is 0.073, and this indicates that 90% of your subjects in your cohort have a calculated risk at or below 0.073, and only 10% have risks above 0.073. So if you're able to do that for, let's say, a population, now you're able to identify at above average risk folks who you would assume would have a higher prevalence of disease. There's also another tool that we could use to identify high prevalence population, and that's something called the progesterone challenge test. So this is basically the PCT test. So this basically assumes that in menopause, which is a period of time or a stage of your life that happens 12 months after periods stop, we expect hormones to fall. And so this low hormone would cause the endometrium to thin. So if there is no estrogen and you give someone progesterone, nothing will happen. But if there is no estrogen, if there is a lot of estrogen and you give someone progesterone, what will happen is it will mimic a menstrual cycle, right? They'll start bleeding. And so what happens in this test is you give someone a short course of low dose progestin for, let's say, 10 days, and then they stop. And if they experience withdrawal bleeding, that means that they have an active or a proliferative endometrium. And if they don't, that means that they have an atrophic endometrium, which is what you expect in a population, let's say, post-menopause. So that's another test that we could use to sort of identify a population of high prevalence of disease. Okay, so now our RESToRE trial. So this is a protocol that we've proposed for screening and prevention. We just recently got funding from the Canadian Cancer Society and CIHR to run this feasibility study. And the objectives here are, can we identify high-risk individuals who will benefit from this screening and prevention in an asymptomatic population? And will these interventions be feasible and acceptable? And can we use minimally invasive tools to detect cancer? So this is the protocol, and I'll explain it. So what we would do is in asymptomatic postmenopausal population of women will be consented, and then they'll take part of our first screen. So our first screen will evaluate an epidemiological score model. And then from there, we will look at, do they fall in the top 25% of risk? And the way we're going to compare the top 25% of risk, and again, is because we do have access to data from the cohorts that these models were developed and validated on. So we will have access to that data, and so we'll be able to sort of see where they fall on the spectrum of risk. Each new recruit, where they fall on the spectrum of risk. So if they're in the top 25%, they are eligible to take part in the second screen. And the second screen consists of the progesterone challenge test. So again, we'll give them the progesterone challenge test. We will look for positive bleeding. And if they have positive bleeding, they go and get an endometrial biopsy. Now, that in of itself, like I've just spent a lot of time talking about why this is, it's painful, it's invasive. We want to try to find minimally invasive tools. So we are going to try also to look at the self-collected tampons and swabs to look for DNA and microbiome signatures. Now, we don't know whether or not the PCT will change what we detect in the microbiome. It would be really cool to only be able to do this as a way to get rid of the biopsy. But because we don't know, we're actually going to collect it before and after and look at whether or not the molecular data changes after the PCT. So that's part of our protocol. And then once they get the endometrial biopsy, if they have cancer, they're excluded from the study because then they would have to get standard of care, including a hysterectomy. But if they don't have cancer, if they have either a proliferative endometrium, EIN, or endometrial hyperplasia, they will get, so if it's EIN or endometrial hyperplasia, they'll get counseled for an IUD insertion as well as hormones or both. If not, everybody will get proliferative endometrium as well as EIN or EH will all get a lifestyle intervention, which is, I've partnered with Dr. Mary Chung, who has the Small Steps for Big Changes lifestyle program at UBC, which consists of 6 sessions of one-on-one exercise and dietary counseling with a trainer over 3 weeks, as well as tracking of exercise with a health app and fitness watch for 6 months. And they will get a complimentary membership at the YMCA. The whole study is going to be monitored, is going to be managed and monitored digitally. So all the symptoms are going to be reported in an app called MyCap, which I'm working on basically developing. It's kind of this MyCap app is associated with the RedCap electronic data capture tool that's very popular and used everywhere in health research. But what this app does is that it's able to schedule various collections, and it's able to prompt daily on symptoms and track basically the participants throughout. It also integrates with the Fitbits, which we will be providing all of our participants. How we will assess feasibility. So again, the primary aim is to evaluate feasibility. We're looking at recruitment. So how many can we screen per month? How many will we enroll? Will there be delays, etc.? We're looking at detection. So how many screen to detect one proliferative endometrium or hyperplasia? We'll look at dropouts. How many will be dropping out of the program and why? We'll look at adherence. Will participants adhere to study protocols? And what are the painful parts of the protocols? Like what are the things that we can improve on or things that we need to fix? And then finally, we want to see whether or not our participants are accepting of the digital health tools and they're comfortable with using those tools, especially that we are looking at a population who is a little bit older, so over 50. For the minimally invasive screening test, we're interested in seeing whether or not we can detect molecular and microbiome signatures associated with proliferation, hyperplasia, or cancer. We're also interested to see whether we can use the tampon test to replace the endometrial biopsy in the screening program. And we want to see whether or not the results are impacted by, the molecular results are impacted by the progesterone challenge test. So this was a lot of information. I've reached the end of my talk. So I want to thank you all for listening. And I want to acknowledge my key collaborators, my team, and of course, my funders. Thank you very much. And I'll take any questions.
Thank you so much. That was really interesting talk, and I mean, quite informative as well. I guess if anybody has any questions, please either raise your hand and you can speak up, or you can type it in the chat and I can read it on your behalf. And I guess I do have some questions, so I'll start maybe with these. So when you're measuring these accuracy statistics, specificity, and like these two main statistics, what is the, I guess, ground truth baseline that you're comparing against? Is it like, yeah, so yeah.
That's a good question. So when we're doing these statistics, again, we're looking at, we compare them to the mutations in the tumor. So these are cancer patients. So we would go look in the tumor and see whether or not the tumor has the same mutation as, and they'd have to be the same clone too. So whether or not they're related. So what happens is the tumor actually sheds through the uterus, and then you're collecting that in the fluid. So if it's in the tumor, you may find it in that fluid. If it's not in the tumor, you're not going to find it, or if you find it, it's not related. And that's what we're
so is it so that the something to be true positive, it's based on having the mutation but not necessarily the diagnosis of the cancer? Is that my is that understanding correct?
So far the way it has been evaluated is that the way that those tests that were done by our team were evaluated is they actually went in a population of cancer patients and they collected, and then they went into a normal population of age-matched patients and they collected. So we don't know whether or not the normals actually they may have cancer, but actually, so the data is starting to come out. So this is very fresh, hot off the press kind of data. We detected no mutations in the normals, and we've detected all of the mutations in the cancer. It's been very stark, which is very interesting. But with that said, maybe again, as we know that with normal mutation, somatic mutations in healthy normals can be found increasingly with age, and there's been several papers to discuss that. And we are expecting, particularly in the population at high risk, that we may find a lot more mutations. And so the test characteristics may not hold once we start going into these high risk populations. So we'll have to see, and that's still something that we're evaluating. Yeah.
Cool. And what is exactly I mean, you discuss different tests, including the tampon tests and the different swabs, but what is the, I guess, the assay after that? Are you doing DNA collection and doing sequencing on these, or is this some kind of is there some simpler PCR based maybe probe or something like that?
Yeah, digital PCR is what we're doing.
So even if the sample collection might be done at home, but still everything needs to be sent to a lab.
Yes, for sure. Yeah. Now, some things you could do, so for example, pH, they could do at home.
Yeah.
But microbiome, and yes, those are all going to need a lab until we figure out a way to do it like a COVID test.
Yeah. I mean, but then I guess even with a COVID test, you can only detect if with some antigen, but not with sequence-based molecular tests. Which, if we have that would be amazing.
Yeah. No, for sure. And again, I think maybe the technology will be there at some point, right? To have a small compute. I mean, a lot of people are looking right now at self-testing and self-tracking is kind of like exploding. A lot of people are looking at assessing hormone levels, for example, which we have another project that's looking at that, because that could also be a factor, like because of these hormonal imbalance or increased risk. And some people could like test their hormones, hormone testing at home. Even though hormones, we need like mass spectrometry to evaluate, that's their gold standard, but maybe there is another test that's not as sensitive or not as accurate, but that gives you generally an idea, and that's good enough, and that's fine. But I think we're not there yet. We're still sort of taking it step by step.
I don't know, I guess we have a couple of points in the chat. Oh, no, there's one question. No, I guess that was just a thank you for the interesting information and best of luck in the upcoming steps. But yeah, there's a question from Vince Montoya. Have there been any studies comparing swabs, biopsies to tampons in terms of microbiome characterization efficiency? If not that, is it surprising if not, then that is surprising and highly unfortunate.
Can I ask you, what do you mean by comparing swabs to biopsies, or swabs to tampons, you mean? Sorry, I'm a bit confused about your question.
Yeah, can you hear me? Sorry.
Yeah.
I'll just ask. Thank you. I just wanted to know, yeah, comparing microbiome efficiencies between the two, like from the tampon versus from a swab, or like you said.
Yeah, so not the microbiome, but we did in our comparison, we looked at the mutations from the swabs and the tampons, and they were very similar, actually. I mean, tampons had a little bit higher allele frequency, but they both detected mutations. So we'll have to see. So this is kind of ongoing evaluation, and that's something we're gonna look at here. We may not have the numbers to do this. Again, I forgot to mention that for the feasibility, all the numbers that we will be looking at, we're probably going to be screening about 600 women. 250 will be eligible for the second screen is what we're estimating. And then from there, we're estimating about 50 will get the intervention. And if we kind of demonstrate acceptability, the next step would be to go into a much larger study. And then that would recruit a lot more. And again, this is still kind of just doing a look-see in this population to see whether or not.
Yes, but even just spiking, like doing sensitivity analysis from like your ability to recover a particular microbe from a tampon to see if there's any like inhibitors or, you know, things like that, that could bias your any microbiome study based on the tampon.
Oh, so in our study, we're doing the tampons for the molecular, but we're doing swabs for microbiome. So they're not the same, they're separate. So, and they need different buffers, they need different media to store it in and so on. And so we're keeping them separate. So there's gonna be a microbiome swab, there's gonna be a tampon, as well as a pH test that they do. So 3 different tests, not in the same test. Does that?
Yes, thank you. Thank you for clarifying. Thanks.
Okay. Yeah.
So we have, I guess, Giuliano is raising their hands, and there's a question from Patty. So maybe Giuliano, you can take it.
Cool. Thanks. Yeah, thank you, Dr. Lin. That was a very interesting talk. I have actually 2 questions. One is more like epidemiological. I wonder, I don't know if I missed this, but I wonder how, like, if mortality or morbidity sort of accompanied the rise in incidence for endometrial cancer, and if like is like overdiagnosis sort of initial in this specific context.
Yeah, so this is all a good question. So like you said, so mortality is following for sure. So in terms of like incidence is rising, and so is mortality, right? Both are rising. Mortality, particularly because it's the more aggressive type of endometrial cancer that's increasing, that we're noticing is increasing. Yeah, so now the other question is, will early detection improve, will early detection improve mortality, or are you just kind of overdiagnosing it, similar to, for example, breast cancer, that now we're really good at diagnosing it, but it doesn't necessarily mean that we're necessarily saving people. And that's a very good point. But I think in this particular population, so we don't know is the answer. So we have to do the studies and evaluate that. But even for, let's say, women with obesity, and when we're talking obesity, like, we're not talking overweight, we're talking obesity, like women with a BMI of 40. Even if you diagnose it, many times these are not eligible candidates for surgery. So surgery is curative in endometrial cancer most of the time, especially if you're detected when it's still confined to the uterus, because all you do is remove the uterus and you get rid of the cancer. But when someone is so large that they're not a candidate for surgery, or the surgery carries comorbid risks and so on, you really don't want them to get the cancer. I think that's what we're after. So you want to prevent it from ever happening in the first place. So because if it happens, you cannot treat them. So there's that a big issue there.
That's very interesting. Yeah, one other question I had, like, so I wonder about like where sort of in the clinical, sort of the patient journey, the screening is going to happen or is supposed to happen, like, because as I understand in the study design, it feels like you're selecting patients based on the PCT for the biopsy.
Yeah.
I wonder if you consider that, like, when you're developing your screening tool, if you might have some sort of like partial verification bias because you're doing the selection based on the PCT for the biopsy, meaning that not everyone is getting the biopsy. Because we can't, of course.
So there may be, okay, so that's the point is you're saying is that maybe the PCT is missing some, right?
Yeah.
They may be missing some patients with cancer. So we're also working, so this PCT test is not new. It's been around since like the 80s and the 90s. And there's been a lot of studies that look at the sensitivity and specificity of that test in detecting cancer. Not cancer, sorry, but cancer, but not just cancer, the proliferative endometrium, really, like it's a spectrum, and it's really high. The specificity, sensitivity, specificity, really high. And the thing is, these are people who are asymptomatic. So in other words, they don't have symptoms. So typically,
so you're not missing
population, they're not missing anything, right? Like, it's not like we're not treating them or anything. It's just like we're going and looking where someone that doesn't have a symptom to try to prevent it. So there may be some that you miss, hopefully not many, but if they're missed, it's no worse than if they were just to kind of not know that they have it and just kind of wait till they get a symptom.
The scale is just so overwhelmingly high. Cool.
Yeah.
Yeah, and if you don't mind, I have like one last question about the feasibility study, about the detection. I wonder if you're considering like sort of the number needed to evaluate, or is like a decision theoretic measure like net benefit?
For which?
For the detection yield in the feasibility study.
Oh, the number that, yeah. So we're looking at, actually, so the number detected, how many, we're just looking at the numbers. And again, because I think the next phase, we will have to power our study so that we could, how many do we need to demonstrate efficacy? And we need certain numbers, and including how many of those that do the PCT will be positive in this population? How many of those that will screen, have a PCT positive? How many of those that do PCT will have an anomaly? So all these numbers are needed to power our next phase. And that's why we are looking for these numbers, right? Because if you want to design a prevention trial to evaluate the efficacy of these preventions, you need to basically get these numbers to to do that, and that's what's needed. And if we can't get those numbers high enough, or if it becomes very infeasible, then we can't evaluate it. Yeah, and I'll have to say too that, you know, the primary reason is the cost effectiveness, right? Like, it's just not cost effective to screen right now, and that's why it's only restricted to populations at Lynch, because they have 10 times the risk of a normal population, and that's why they get screened, and also women with abnormal bleeding, because they're at higher risk. So that's why I think the importance of finding these high-risk populations, then you could justify screening them, right? And the other thing that I wanted to say in terms of efficacy of the interventions, the program, the Small Steps for Big Change, is a program that's conceived for pre-diabetics. So it's also, and it's shown to be effective there. So I feel like, you know, we already have a lot of tools that are in place to sort of demonstrate this.
So I have like indirect evidence, like for similar.
Yeah, exactly.
That's very cool. Is it in your roadmap to do a randomized clinical trial for this screening?
So it won't be randomized, but it would be, there will have to be a clinical trial after this, and that would be part 2 of this project.
Very cool. Very cool. Thank you. Thank you so much.
Thank you so much, Giuliano. We do have a question also in the chat from Patty Ye, and it is, has there been a prevalence in EC in transgender males that are undergoing hormonal therapy?
So we don't know about EC, but we know that transgender male, so anyone with a uterus is at risk. So if they still have, if they're transgender male and they're they haven't undergone the surgery to remove their uterus yet, then they would still be at risk. And we know specifically, there was a study about polycystic ovarian syndrome, which is a risk factor for endometrial cancer. And we know that there's a high prevalence of PCOS in transgender male. And it often, so we are not excluding them. So anyone that is transgender male would be part of the study.
How does, I mean, I guess, yeah, like you've mentioned, like anybody with a uterus is at already at a risk factor because it is a uterus-based cancer. But does hormonal therapy exaggerate or maybe undermine the risk for?
Totally does. It totally does. Yeah.
Sorry, in which direction?
It increases. So it depends. So generally speaking, if you're taking, again, it's an estrogen dominated disease. And so if you have estrogen and not enough progesterone, so when does that arise? It could be in premenopause, when a woman is still producing but not producing enough progesterone. So that could happen, for example, if they don't ovulate. And if you don't have ovulation, the uterus doesn't get the signal to start producing progesterone. And so, because there's not going to be a pregnancy, and so there's excess estrogen. So that's a problem. It also PCOS is another problem that kind of causes this in premenopausal women. Postmenopause is generally obesity. Transgender male, if they're taking too much estrogen hormones, that could also be a problem, right? So especially, again, depends on each person, their own physiology could also impact things as well. So, and exogenous stuff, right? Like we haven't talked about exogenous stuff, but there's also all sorts of things in the environment, using PBA, right? Like there's all sorts of things of exogenous. Women who get breast cancer at an increased risk because of the secondary effect of their tamoxifen that they take as a treatment for their breast cancer also produces estrogen. There are some tumors in the body that produce excess estrogen. The tumor itself may be benign, but it could lead to endometrial cancer as well. The other thing about endometrial cancer and why it's important to intervene, kind of intercept it, I often feel like it has a very good survival rate, like 95% survive, but 50% of those that survive the cancer will go on and die of causes that are associated with obesity and diabetes and things like that. And so, in a way, it's almost like preventing the cancer not only prevents death of cancer, but it also prevents the death of other causes, especially if you worked at reversing these risk factors, right?
Cool. I do have one more question, and I guess if we don't have any others, then we can wrap up for today. My question is about going back to like the questions of like sensitivity of these different tests. Do you think that they are systematically biased? The error is systematically biased. So if you do repeat the test multiple times, can you like even get to like 100% accuracy, or do you think there's like some systematic bias that multiple tests would still produce the same error?
I don't understand your question very well. So what do you mean?
Oh, so like the tests, for example, like the tampon test, it has some error, and like if you do it like multiple times, do you still expect like you can completely eliminate that error, or no?
I got it. Yeah. I mean, so the way, again, we're still not sure, right? How you'd evaluate. First of all, we need to make sure that currently we just kind of developed the protocol. We tested it in a population that's completely, it's not like tested in the wild in a way. It's kind of in a study that's very kind of, we're selecting the populations. It's not really going and testing it on a group of unselected participants. Which this study may do, right? Because this we're not selecting. We're just kind of looking at risk factors, and if they are high risk, they could come in. And now, if you look at the numbers may change, and I suspect they will. That's the first thing. Second thing is, how will this be implemented in practice? Like, would the test be, if the test is only kind of prompting a secondary test that's more specific, like a biopsy, for example, right? Then it doesn't matter if you make an error. Again, if you miss it, if there's a mutation and you've missed it, it just means that they're going to get delayed. I mean, I guess it depends if they have symptoms or not, but if you're thinking asymptomatic population, again, if the test misses a mutation, it doesn't matter. If it doesn't miss the mutation, detects a mutation, maybe at first, you have to see whether or not the mutation is also detected in the biopsy. But say, no, if I detect any mutation that's persistent over time, so maybe you detect the mutation now, you look at in 6 months and it's still there, the same one, right? Yeah. Now you become more suspicious and you say, okay, this is real and it's not going away. Let's intervene, right? We still don't know how it's going to be used. Right now, we're still sort of doing the proof of concept to see whether or not it can be used at all. Yeah.
Okay. Well, thank you so much. And I want to thank also Angela for your talk, and thank you, Dr. Aline, for the great talk, and for the audience. And that's going to be a wrap for today. Thank you so much.
Thank you.